For the Institute of Systems Genetics at NYU Medical School. He's a member of the National Academy of Sciences and the American Academy of Sciences. And as his name alludes, he's really been a pioneer in the field of high-throughput genetics. While he was here at Johns Hopkins, he was part of the yeast deletion collection project, where him and his collaborators were among some of the first researchers to study genetic interactions between the thousands of genes that were uncovered in the sequencing of the yeast genome, and really start to get at what the function of those genes were. And since then, he's undertaken an even larger project, in my opinion, which is to understand the function of structural variation in genomes, and which is, of course, much more difficult with traditional approaches. So Jeff's really taken a new approach, which is to make his genomes himself. And along with his collaborators, they've been synthesizing yeast chromosome by chromosome, rearranging them, deleting repetitive elements to try to study what the function of all this structural variation is. Ever since he was a graduate student with Gerald Fink, he's been interested in one type of structural variation that Julia showed us is really rampant in all genomes, including ours, which is that of transposable elements. In 1985, Jeff was the first to discover that a yeast transposon called TY, actually using a genetic reporter, showed that it hops throughout the genome through an RNA intermediate and reverse transcriptase, just like retroviruses. And since then, he's really made a lot of progress in uncovering the mechanism of retrotransposition, which is a term he actually coined, and also how transposons regulate themselves and their activity within cells and organisms to become such a successful parasite. And so today he's going to tell us about one mechanism that line 1 does this within our genome.
Okay, thank you very much, Steve, and thank you very much for inviting me back to Baltimore. When I was a faculty member across town, I always loved coming to these mini symposia, so it was an honor to be invited to speak at one of them for the first time. So all of you local Baltimoreans will recognize the iconic image in the background here, and I'll say a little bit more about that towards the end. The significance of it will become clear. I am going to talk primarily about line sequences, and one of the changes that happened when I moved to my new institution was that I was required to, let's see here. Oh, I guess I just use this. Okay. I'm required to show the logo of my new institution. It's kind of a formal place, and but being a rebel at heart, I insist on printing the logo using engineered recombinant yeast that produce pigments of various types. So that's also what you see in the background here. Okay, so so retrotransposons are everywhere in biology, and they make up a massive proportion of our genomes, and they are really adaptation machines. What you see here in this phylogenetic tree is the proportion of various types of genomes, ranging from microbial genomes, which are super streamlined and have relatively few transposons. There are some there. The slice is just too small for your eye to see. To mammalian genomes, which are heavily dominated by retrotransposons. So in the mouse and human genomes, we see these very large slices in red, which represent the line sequences that I'll talk about today. So these are present in hundreds of thousands of copies in our genome, and this represents kind of a look back in evolutionary time, because most of these copies are rusting hulks that transposed long ago in the genomes of our ancestors. They're no longer active, and only about 100 copies on average in a human diploid genome are actually active. So obviously, with this kind of interaction with our genome, we we've seen the evolution of this arms race that you just heard a beautiful story about, and that's really just one of many mechanisms. We'll hear more from Harmeet about this. Many mechanisms that have evolved over the years and in different systems to control the proliferation of these retrotransposons. So we've also been very interested in this sort of host-parasite interaction and some of the things that might regulate it. So I've recently been pondering how was it that I kind of made this career twist and turn from studying reverse transcription and retrotransposons to actually writing down new genomes. And I realized that I had unwittingly taken inspiration from retrotransposons. So of course we all know that DNA makes RNA, and retrotransposons and retroviruses use reverse transcriptase to make DNA from RNA. And many other people have pointed out the likelihood that we started out with an RNA world and it went to a DNA world at some point. DNA being a much more stable form of archiving information. And therefore, to make that transition, you would have needed reverse transcriptase. And this is, I think, a powerful argument that reverse transcriptase is probably one of the most ancient enzymes there is. It's probably derived from RNA polymerase and is in fact the original writer of biological genomes. And so in fact, I created this figure when I was in Jerry Fink's lab. It was fanciful at the time, but I actually think this is quite true, that we can think of most of the DNA in the genome as arising from ongoing cycles of transposition, degeneration of mobile element sequences into pseudo-random sequences, and eventually being selected upon for new functions. And this is probably one mechanism by which genes were born. So in our genome, we have at least 50% repetitive DNA, and of that, and that's a conservative estimate. So de Kooning et al., using a more sensitive method for detecting ancient copies of repeats, suggests that it might be as high as 78%. So certainly the vast majority of our genome has been written by reverse transcriptase over the years. And then my colleague Kathy Burns across town and others have shown that in a subset of human tumors, this enzyme very actively produces new copies, and there are these somatic insertions that occur in cancer cells. Other work has shown that it also occurs in senescent cells and in aging tissues. These elements become de-repressed and produce lots and lots of new copies. So suggesting that this is something that our National Institutes of Health should pay attention to and perhaps invest in. Okay, so so what we've studied really over the last 20 or 30 years is this process of retrotransposition. So here you see the anatomy of the line sequence, sometimes called line 1, sometimes L1. It has a peculiar promoter structure where it's a Pol2 promoter, but all of the elements that control the expression are downstream of the transcription start site. And then, as is the case with many Pol2 promoters, there's an antisense promoter that fires off to the left and reads into adjacent cellular sequences. There are no introns, and there are two open reading frames. The first one, ORF1, is a very abundant protein, and it's an RNA binding protein that forms, binds to the line 1 RNA and forms a ribonucleoprotein particle. The second open reading frame is the reverse transcriptase and endonuclease bifunctional protein. And both of these proteins are essential for the process of retrotransposition. At the 3' end, these peculiar genes also differ from typical Pol II transcription units in that they have poly A actually in the DNA. These are really unusual transcription units that, of course, have the ability to move themselves around. And when they insert, they generate a variable length target site duplication. That's an unusual feature of this class of mobile element in that the length is variable rather than fixed, as in most transposable elements. So the life cycle starts out with production of RNA by RNA polymerase II. That is exported through a mysterious mechanism into the cytoplasm where translation occurs and the ORF1 protein trimers and possibly just a single molecule of ORF2 protein bind to their own messenger RNA. It's a cis-acting process. And then this is re-enters the nucleus, this RNA, in the form of the RNP, where it carries out a process called target-primed reverse transcription. So the endonuclease makes a nick in a new target site, and then that nick is extended using the line 1 RNA as a template, and then there are subsequent steps that are much less well understood mechanistically that lead to new insertions, and the new insertions are often severely truncated. So that's the basic life cycle. We've been very interested in host factors that control these various processes. And so today I'll talk a little bit about some of the factors that bind to the promoter, and I'll talk a bit about the nuclear import and export and really when in the cell cycle this blessed event here, the TPRT, target primed reverse transcription, actually occurs. Again, my colleague across town, Kathy Burns, has done extensive work on line element expression, and interestingly, about 50% of human tumors show very high levels of expression of the ORF2 protein, and this phenotype is highly correlated with P53 defective tumors. Those are the ones that really overproduce the elements, and so we know that she has some other very exciting evidence that indeed P53 is very responsive to transposons, and when it's defective, you see this massive overexpression. Massive overexpression is necessary but not sufficient for retrotransposition. So whereas 50% of tumors overexpress the proteins, not all of those show high levels of somatic hopping. And other work from my colleagues at NYU, Shuya Wang and David Fenyo, show that indeed the vast majority, for example, of breast and ovarian tumors produce ORF1P directly detectable by mass spectrometry. So I'm going to just give you the highlight of a study that we just published in PNAS from Sunny Sun and Shuya Huang, where we basically mined the ENCODE data set to study what everybody else ignores, namely the sequences that got masked and where transcription factors that people have been mapping the binding sites, where they are binding to the line sequence. So what you'll see in these graphs is the consensus L1HS. This is the highly active young elements that are in your genome and jumping as we're sitting here in your genome. that element, the consensus element, is about 6,000 base pairs, and this region here is that 5' untranslated region / promoter of about 900 base pairs. And this just shows the trace that you get from the MYC oncogene. And you can see that there are 3 huge peaks in this particular cell line binding to that promoter region, and a smaller peak in the 3' untranslated region. And one of the very interesting findings in this paper was that there's a second type of motif. There's the MYC motif itself that lines up with some of these peaks. But there's another peak called disk 10, which has been associated with MYC binding, but is actually a CTCF binding site. And it turns out that MYC and CTCF cooperate to bind in many places. CTCF, of course, being the very important architectural protein responsible for the formation of transcriptionally active domains. And so we can imagine that this element then has the potential, when it inserts into a new location, to change chromatin architecture and potentially change the structure of topologically associated domains, or perhaps even create its own mini-TAD. So this came out of a larger experiment to do what I call drinking from the fire hose. We searched an unbelievable number of data sets in the ENCODE, every human transcription factor and chromatin data set that was deposited. We wrote a pipeline called MapperCon for that basically maps reads, deep sequencing reads from ChIP-seq to a consensus sequence for retrotransposon of interest, in this case line 1. And then this gives you this very nice set of peaks. And one of the really amazing but slightly daunting results from this was that 32% of transcription factors that had been tested by the larger ENCODE community showed very clear binding peaks, and the vast majority of those are in the promoter region of L1, but some of these factors bind very specifically to internal regions of the line 1 genome. And so MYC was one of those that came out of that. All right, so that's all I'll say about that. Obviously, you're welcome to take a look at the paper if you're interested in seeing more on that. The main topic for today that I wanted to talk about is some other work that was very recently published in eLife by Paulo Mita and others, which has to do with the question, is the process of retrotransposition random, or is it somehow associated with the cell cycle? And to get a good answer to this question required the development of some really good reagents. So Paulo became quite expert at looking at the proteins, both the ORF1 and the ORF2 proteins, and developing tools for following those throughout the cell cycle. And so what you can see here is that the ORF1 protein is in most cells clearly present in the cytoplasm, often in a perinuclear location. But if you look carefully, you'll see that in some cells, you'll see some ORF1 protein in the nucleus, and in other cells that is much less clear. And so you can see that when you do see ORF1 protein in the nucleus, you also see ORF2 protein in the nucleus of those cells. But after cell division, during the G1 phase, the ORF1 protein disappears from the nucleus, And this appears to be, at least in part, through an active export process because it can be blocked with the drug leptomycin. So Paulo was able to show that in the G1 phase, the nuclear association of ORF1 became very obvious in the G1 phase. And then as cells transitioned to the SG2 phase, this led to a loss of ORF1p from the nucleus. So you can see, hard to see here. You can see the cells with the red arrows have the CDT1 staining, so those are the G1 cells, and those are the ones that actually have the nuclear ORF1p, whereas these with the blank nuclei, those are all, with the green arrows, they have very low CDT1. And so using CDT1, a G1 marker, and geminin, which marks the other phase of the cell cycle, it was very clear that this was the trajectory. So what we infer from this is that the ORF1, which is, of course, the main marker we have of the ribonucleoprotein particle, probably entered the nucleus during the prior mitotic phase when there would be nuclear envelope breakdown. And then once the RNP was delivered to the nucleus, or perhaps delivered to chromatin, the ORF2, for reasons we don't really understand why, ORF1 is released and actually exported from the nucleus. That's our conclusion from these experiments. If we look at where the proteins go in a chromatin prep, we saw that while there is some association of ORF1 protein with chromatin, it's fairly flat across the cell cycle. But if we look at ORF2, we see a clear peak during the S phase at the same time that PCNA associates with chromatin. And some of our earlier work had shown that PCNA interacts directly with ORF2 protein. There's actually a PIP box PCNA binding sequence between the endonuclease and reverse transcriptase domains of the ORF2 protein. So, so this was one thing that pointed to the likelihood that retrotransposition, the actual TPRT step, might be occurring during the S phase. The protein that carries it out is on chromatin during that period of time. But we wanted to do some experiments to really try to look at when the retrotransposition event was occurring. And so the first experiment that Paulo designed to look at this involved a rather complicated set of treatments where we had the element under the control of a TET promoter. We would turn on the expression, then we would arrest cells with nocodazole. We would shake off the arrested cells and wash out the nocodazole, and then follow these cells over a period of time. And these, the transposon in this case is tagged with a GFP marker that becomes activated upon retrotransposition via the intron removal that occurs in the transposon. So, so in other words, once you see a green cell, you know that it must have transposed within the past several hours. And if there is no effect of the cell cycle, then you would just expect these green cells to accumulate linearly over time. Whereas if there was a cell cycle component to when it was happening, you would see a nonlinear result. And by taking the first derivative of these, you might see a peak that would tell you when you could sort of walk backwards to infer during what period that would have occurred. And so, indeed, what we saw in this experiment was that when we took the first derivative, we saw the peak GFP rate of GFP accumulation in the late S phase, and then by accounting for the amount of time it would take for the GFP to become expressed and so on, we inferred that the transposition would have taken place a couple of hours earlier in the S phase. Okay, but this is a messy experiment using a lot of drugs and manipulations, and so it would be nice to have a more direct and independent validation of this conclusion. So Paolo's really great idea was to employ this very useful tool called the fluorescent timer, which is a GFP protein that starts its life out, the first 2 to 3 hours of its life, it's a blue fluorescence, and then it transitions to a red fluorescence fairly rapidly after that. And so this allowed him to look at this process in a pool of unsynchronized cells, which were then sorted and could be sorted on either the blue, the red, or the intermediate shades. And the result was really quite spectacularly clear. When blue cells were sorted, there was a huge peak in the S phase cells, whereas when the red cells were sorted, they were all in G2M. So, again, accounting for various timing aspects, we were able to infer from this that the transposition event, the actual TPRT event, occurred during the early S phase, well, S phase, probably early S phase. In a second approach, he took a affinity proteomics approach by pulling down on the ORF2 protein that was bound to chromatin. And so we used a flag-tagged ORF2 and compared the pull down of ORF2 IP followed by a PCNA IP, and we of course pulled down PCNA, but we also pulled down a number of other replication fork components. So this is a piece of evidence that suggests to us that not only is it in the nucleus during the S phase carrying out the hopping, but it actually seems to be associating with some replication fork components. And finally, in some cells that were arrested in the S phase, he could actually show that a subset of PCNA foci were associated with ORF2 protein, again putting these two complexes in the same place at the same time in S phase. So putting together all of these facts, we conclude that something like this model might explain our observations. So when we have line 1 expressed in cells, we get these ribonucleoprotein particles shown here as orange dots representing ORF1 protein and green dots representing ORF2 protein on the RNA. During mitosis, a subset of those get access to the nucleus. In the daughter cells, we have those particles present both in the nucleus and in the cytoplasm. In the later G1 phase, we see the loss of the ORF1 protein. We don't know why that would be, but it might reflect some chaperone activity of ORF1 that its job is done and is no longer required. And then I'll show you some other evidence that there is definitely a separate form of the RNP that consists just of the ORF1 protein and the RNA and doesn't contain any ORF1 protein, ORF2 protein and RNA and no ORF1 protein. And then during the S phase, these complexes perhaps target replication forks, maybe stalled replication forks, and this is when the actual retrotransposition occurs. So a second study that we did in collaboration with John LaCava that very much supported this was an extension of our earlier work where we developed these tagged retrotransposition reporters in which either the ORF1 or ORF2 proteins were tagged in such a way that didn't interfere with retrotransposition. And so these constructs in red, you'll see, show very high levels of retrotransposition despite the fact that the proteins have been tagged. So we know that they're functional and active. John LaCava, Marty Taylor, and others developed this system where we can grow large quantities of cells in suspension culture, grind them into very small particles in liquid nitrogen, and then do a very rapid pull down with the affinity tag on magnetic beads and get very clean preparations for mass spectrometry. And in this second paper by Taylor et al., what we showed was that we could look, for example, at a pull down and then treat the pull down with RNase, or do a second treatment with anti-ORF1 to remove any ORF1. And what we showed was that there were two different forms of RNP, one of which is highly enriched for cytoplasmic proteins and is associated with particles that have abundant ORF1 in them, and a second cluster that's associated with nuclear factors and is very much depleted of ORF1. So this is some of the other evidence we have to suggest that there are at least two distinct types of RNPs in these preparations, and it's very consistent with the work that Paolo did showing the cell biology. So this just shows some of the factors that are associated with both the cytoplasmic and the nuclear forms of the RNP, and some of the roles we think they might be playing in the different steps. So one last piece of evidence that cemented the idea that the replication fork might be a target came from a high-throughput screen that we did for siRNAs that inhibit basically siRNA pools against every human gene, and the cells were transfected with one of these L1 GFP AI reporters. And then we use a microscopy-based scan to look at the frequency of green cells. And what we found at the very top of our heap was a cluster of proteins that resulted, when knocked down, resulted in a very much higher level of retrotransposition. So these would be potentially control factors that are preventing retrotransposition. And the number one hit was actually BRCA1, and several factors that interact with BRCA1, which are the Fanconi anemia proteins, which are involved in repairing stalled replication forks, are all in this cluster of top hits. So this is one more piece of evidence that leads us to suspect that the replication fork itself is the target. So this just shows some of the hits that came up in our screen, and that they're well-known cluster of proteins that work together. So what might this mean? Well, it might mean that the line sequences are somehow identifying these stalled replication forks and preferentially inserted there. Or I think an equally attractive hypothesis is that they insert, and actually the active insertion creates a blocked fork. And then the Fanconi anemia factors may act to restore the blocked fork and perhaps degrade the partially retrotransposed, the partial retrotransposon insertion. So this is something we're really looking into now. So the question then comes up, why would this cell cycle regulation occur? So yeah, maybe L1 prefers to target replication forks. We know that PCNA behaves like a host factor for L1. Maybe this is a phylogenetic memory of an ancient endonuclease-independent type of element described by Belfort and Lambowitz. These are the group 2 intron reverse transcriptases. They've been shown to target the lagging strand where there's a pre-existing 3' end that can be used in a TPRT reaction. So that may be one origin of all of this. Kunin has hypothesized that as the host genome grew due to constant invasion, this eventually evolved into the nucleus as a firewall against mobile elements. And so, so perhaps it's using a window of opportunity right after mitosis to get into the nucleus. And I just heard a new hypothesis from Alan in the introduction, which was that if you transposed outside of S phase, that might provide a signal that you're an invader, so that would be a bad thing. And finally, I like to call the man's got to eat hypothesis. So a transposon, a retrotransposon's got to eat a lot of triphosphates. And so it's actually well known that triphosphate levels are very tightly controlled, probably as a defense against retro elements, to be high only during the S phase. And so there are many proteins that control this, including SAMHD1 in humans. When SAMHD1 is knocked out, you have much higher triphosphate levels throughout the cell cycle, and you also see overactivation of retrotransposons. So that's all I'm going to say about transposable elements in humans, but I can't resist saying just a few words about yeast. Steve alluded to this in the introduction, that we've been building these synthetic chromosomes, and we've systematically exterminated every base pair of transposable element from these chromosomes. And one of the things we were quite fascinated about, and I think one of the most interesting results from our recent set of papers, is that the trajectories, the 3D trajectories of native chromosomes and synthetic chromosomes that lack those TEs are pretty hard to tell apart in 3 dimensions. So it was a little bit disappointing as a transposon biologist that, you know, the transposons didn't do anything in yeast. And this was even true if we would combine two synthetic chromosomes together. We still saw quite similar trajectories. But I do think that this kind of building genomes from scratch can be a very powerful tool for us to probe the basics of genome biology. And I will leave you with a few last experiments from an amazing PhD student who asked the simple question, can we fuse yeast chromosomes together into giant chromosomes? And so she came up with a clever trajectory of fusing chromosomes together two at a time, developed a CRISPR-Cas-based method to basically target two telomeres together and delete one centromere all in one step, and came up with this way to get down to 4 roughly equal-sized chromosomes, and then 3, 2, and perhaps 1. And the answer is yes, we can fuse yeast chromosomes together. Here's a pulsed field gel, and you can see the chromosomes disappearing from the bottom of the gel and growing. We had to change the gel system because the chromosomes were getting so big. And just a few months ago, she had her N=2 yeast born. And these strains are remarkably healthy. They do have a couple of amino acid substitutions that might be providing some suppressor effect, but they have remarkably high fitness considering the fact that they've gone from 16 chromosomes down to 2. And this shows a 3D rendering of the N=16 strain from our colleagues in the Pasteur. And this shows 3D rendering of the inferred structure of the N=3 strain. So you can see the Rabble orientation of the centromeres here are at the top, and the arms coming down in a nice smooth V shape in the wild type, and in the N=3, it's kind of a really gnarly looking structure that's wiggling back and forth a lot, and the centromeres seem to be held together much more tightly than in the N=16, and the telomeres are also forming one tight cluster. So we're very, very excited about this, and we're thinking about all kinds of cool experiments to do with this, so I couldn't resist showing you that. And gratuitous, irrelevant slide on yeast art just because we can use robotics to fly tiny droplets not into a well, but onto an agar plate. We have yeast into which we've built chromosomes that specify the formation of lots of different pigments. And so if you're interested, have a look at yeastart.org. This is where our natty bow came from. That is a 384 biopixel first drawing, which was done by an engineering student here, Jerry Wang. And we're now up to 25,000 biopixels in some of the more recent artworks. So with that, I will show you our trailer for our forthcoming yeast movie. And thank very much the L1 group here that did the work and our remarkable collaborators. Thank you very much.
Hi. So in your story of the line 1, you used the HeLa cells, which is the cancer cell line. Have you tested this in the other cell lines like the germ cells, or do you have some speculation of whether it will be similar as in the cancer cells?
Right. We have not done very much yet with real germ cells. This is also talking about work of others. They're active in embryonic stem cells. They will retranspose in embryonic stem cells. That's work of Gerald Schumann and others. But the work on the cell cycle and so on hasn't been done in those instances. Wenfeng An has been studying the retrotransposition in the germline in the mouse, but again, hasn't been able to look at the cell cycle aspect, as far as I know.
So there's a recent work from Joanne Waisaka's lab showing that you have the derepression of these L1 retrotransposons in response to the Fanconi anemia and some of the homologous recombination pathways. They were looking at expression though, rather than activation. So I was wondering how that might work, how you would get the increased expression of these retrotransposons based on your collapse replication fork model.
They actually looked at both things separately, and my understanding of the paper was the Fanconi anemia effect was only on the hopping, not on the expression.
Okay, so it was just the RAD51 was on expression. Okay, thanks.
Hi. So during the S phase, when some of the particles get trapped in the nucleus and they lose the orphan-P, do you think that the line 1 RNA could be used using homology to recognize specific regions? So could you see something like RNA-DNA hybrids? And could you map the sites where it inserts? So does it insert through TPRT during that stage as well?
So if I understand your question, you're asking whether the RNA in the RNPs is targeting new insertions via sequence homology. We haven't directly looked for that because it's kind of a hard thing to do. But my guess is that it's probably not a big factor because the thing that initiates TPRT is the endonuclease cut. And while those sites are certainly present in inline sequences, they're present everywhere. The consensus sequence for it is very degenerate. It's basically a 6 base pair, all A's and T's, so it comes up. And it's not even it's just a consensus. It's not an absolute requirement. So, so I can't rule it out, but I haven't seen any evidence that the RNA is directing the targeting.
So my question, Jeff, is it's really interesting that transposition happens in S phase. You think it's because of increased NTPs. My question really is, how do you protect the genome? Is there something special that you actually do in S phase that could really kind of control the transposition frequency?
So what we think actually is that this Fanconi anemia complex is actually, through its resection activity, might be able to reverse some partially reverse transcribed TPRT events and essentially, reverse the process before it's gone too far, gone to fixation. Because if you think about it, the reverse transcriptase, it has to go for hundreds to thousands of base pairs of reverse transcription. We don't really know exactly how long that takes, but it's going to take some amount of time. And so if the Fanconi complex can disassemble the fork and into the adjacent DNA, it might be a way to reduce the number of transposition events that go to fixation. That's the idea.
So it's a super, super cool experiment to put this mega chromosome, the 2 mega chromosomes together. Have you seen, so the question, of course, the obvious one is, why do we have more? What's the advantage? Have you seen anything in growth rate or any first signs what would be sort of, altered?
Right. So in the N=2 strain, there is a slight growth defect. But it's like, in a competition experiment, it's like 96% as good as wild type. So you have to work hard to see it, okay? But there is the possibility, and I'm hedging a little bit here because we don't have definitive answer on this point. There's one mutation that arose at the N=14 stage and it's in a protein that controls mitotic exit. So it's kind of suspicious looking, and it's a missense mutation. It's predicted to be deleterious. And that may be some kind of a suppressor mutation. So just keep that in mind, that's a caveat. But from just to what I think in terms of, we have this bigger question, right, of why do we have these vastly different numbers of chromosomes in different organisms? And Saccharomyces cerevisiae, it's a generalist species. It does well in a multitude of different environments. And I believe that it probably got, that 16, which is the number that's in all Saccharomycetaceae, more or less, it's not a random number, okay? So you could imagine going from 1 chromosome to 2 to 4 to 8 to 16. And we already know that there was a whole genome duplication that went from 8 to 16. So there's another big group of pre-duplication yeasts that have a modal number of n equals 8, another one that has n equals 4. And those n equals 4 strains are very fastidious. They're not generalists at all. So I think to be a generalist, you want to be able to adapt quickly. And one way to adapt quickly is by duplicating a small chromosome and increasing the number of gene copies for that chromosome. And this has actually been shown to happen by Pilpel in adaptation to high temperature. You get duplication of chromosome 3. So those kind of duplications would be much more likely to occur when you have a larger number of chromosomes. So that may be one thing that's driving this. The second thing is, accidents of history, like this whole genome duplication and so on.